So I hope the video that you just saw, it's helpful to helping you understand and visualize how the process actually operates. Here is a simple overview of the basic functions and jobs of each of the respective organs. Now, what I want to share with you now is talk a little bit about how does life actually begin? How does the organization form after fertilization has occurred? One of the first things that has to happen before any organism or new cells could be formed is you need to get the genetic material from the sperm and the egg to unite and come together, forming a diploid set of chromosomes. Here is a photograph of what a mature ovum actually looks like. What you're seeing right here is that you are looking at that there are two layers that surround the genetic material that is inside the nucleus of a cell. Here's an area called the zone of pellucida, and then there is also the corona radiata. Now, this is an image of what the egg cell looks like, which is this large sphere, and surrounding it are actually thousands of sperms all around itself. What happens is right after when the sperm makes its way into the female's body, it has to choose either left or right because there are two tubes. There's a right fallopian tube, and then there is the left side fallopian tube. One of these tubes, because typically females only ovulate one egg, only occasionally would the person be ovulating two eggs at once. One of the tubes will be empty. The other one is where the mature ovum is sitting and waiting for a sperm cell to come in. All of these sperms would be swimming, competing against each other to find the first round object and then try to fertilize it. Now, sometimes mistakes would occur, such that the sperm cell would be fertilizing the first round object that it finds, and that may be just a regular body cell, not actually a sex cell. And then in other cases, it would find an empty tube, in which case nothing would come to it. And after about 24 hours sitting inside a female's body, the woman's body would push it back out and it would be disintegrated. But on the other hand, the sperms that finds the tube that actually contains the egg, they actually have to compete against one another to find its way to get to the egg. And right after when they get to the external portion of the egg, which is the corona radiata, it would have to use the digestive enzymes, which are contained on the head of the sperm, known in a region known as the acrosome, and find this and digest this way through this layer until it can enter to the center of the nerve cell of the egg cell and release the genetic material that is stored inside the head of the sperm, allowing it to penetrate and combine with the 23 chromosomes that are sitting inside the egg cell awaiting for fertilization to occur. The midpiece and the flagellum actually does not enter the inside of the egg cell. It stays outside, and the moment that diagram E actually occurs, then the eggs will immediately make a chemical change such that none of the other sperm cells that are competing for entry would be able to penetrate or be allowed to enter the egg cell. Here, in this particular diagram, the reason why I like it and I want to show you is that you can actually see that the chromosomes that has been formed from the second meiotic cell division, you probably recognize this as being in metaphase 2, they are lined up in single file, they are double-stranded, they are actually waiting in that particular position, suspended inside the egg cell until a sperm cell comes in to fertilize with it, and that they would then come together and realign themselves, forming a normal diploid cell, allowing the next step, which is fertilization, and then following that mitosis to occur so that the body of the baby could be formed. Let's take a look at how this actually happens in a video clip.
Fertilization is the epic story of a single sperm facing incredible odds to unite with an egg and form a new human life. It is the story of all of us. During sexual intercourse, about 300 million sperm enter the vagina. Soon afterward, millions of them will either flow out of the vagina or die in its acidic environment. However, many survive because of the protective elements provided in the fluid surrounding them. Next, the sperm must pass through the cervix, an opening into the uterus. Usually, it remains tightly closed, but here the cervix is open for a few days while the woman ovulates. The sperm swim through the cervical mucus, which is thinned to a more watery consistency for easier passage. Once inside the cervix, the sperm continues swimming toward the uterus, though millions will die trying to make it through the mucus. Some sperm remain behind, caught in the folds of the cervix, but they may later continue the journey as a backup to the first group. Inside the uterus, muscular uterine contractions assist the sperm on their journey toward the egg. However, resident cells from the woman's immune system, mistaking the sperm for foreign invaders, destroy thousands more. Next, half the sperm head for the empty fallopian tube, while the other half swim toward the tube containing the unfertilized egg. Now, only a few thousand remain. Inside the fallopian tube, tiny cilia push the egg toward the uterus. To continue, the sperm must surge against this motion to reach the egg. Some sperm get trapped in the cilia and die. During this part of the journey, chemicals in the reproductive tract cause the membranes covering the heads of the sperm to change. As a result, the sperm become hyperactive, swimming harder and faster toward their destination. At long last, the sperm reach the egg. Only a few dozen of the original 300 million sperm remain. The egg is covered with a layer of cells called the corona radiata. The sperm must push through this layer to reach the outer layer of the egg, the zona pellucida. When sperm reach the zona pellucida, they attach to specialized sperm receptors on the surface, which triggers their acrosomes to release digestive enzymes, enabling the sperm to burrow into the layer. Inside the zona pellucida is a narrow, fluid-filled space just outside the egg cell membrane. The first sperm to make contact will fertilize the egg. After a perilous journey and against incredible odds, a single sperm attaches to the egg cell membrane. Within a few minutes, their outer membranes fuse and the egg pulls the sperm inside. This event causes changes in the egg membrane that prevent other sperm from attaching to it. Next, the egg releases chemicals that push other sperm away from the egg and create an impenetrable fertilization membrane. As the reaction spreads outward, the zona pellucida hardens, trapping any sperm unlucky enough to be caught inside. Outside the egg, sperm are no longer able to attach to the zona pellucida. Meanwhile, inside the egg, the tightly packed male genetic material spreads out. A new membrane forms around the genetic material, creating the male pronucleus. Inside, the genetic material reforms into 23 chromosomes. The female genetic material, awakened by the fusion of the sperm with the egg, finishes dividing, resulting in the female pronucleus, which also contains 23 chromosomes. As the male and female pronuclei form, spiderweb-like threads, called microtubules, pull them toward each other. The two sets of chromosomes join together, completing the process of fertilization. At this moment, a unique genetic code arises, instantly determining gender, hair color, eye color, and hundreds of other characteristics. This new single cell, the zygote, is the beginning of a new human being. And now the cilia in the fallopian tube gently sweep the zygote toward the uterus, where he or she will implant in the rich uterine lining, growing and maturing for the next 9 months until ready for birth.
I hope you found that video clip exciting. I was particularly drawn by the visual images of how the egg cell and the sperm cell's genetic material unite with one another by forming those pronuclei. Now, so what happens if the mature ovum does not get fertilized or if it's not used at all? Well, that's where the menstrual cycle begins. So if you take a look at this diagram, you will notice that on the lower part of this diagram, it represents the endometrial cycle and the endometrium, which is the lining that is right above the uterus itself, all right? And it shows how the uterus lining thickens over the span of a month. Um, at the early phases of the cycle, here is the egg follicle, and it undergoes the meiotic cell division such that it completes it, and then the follicle becomes mature. It gets ready to have its egg cell being released. And if you remember from the uh, previous video clips, you saw how the fimbriae is stimulated by chemicals that are released by the follicle itself, such that it would be those projections would then be moving towards in search for this particular egg cell. Once the egg is released, we say that ovulation has occurred. It will then be finding its way into the fallopian tube where cilia will propel it along the way. Now, during this process, there are a number of changes that happen on the thickness of the endometrium. You will notice how the amount of tissue increases inside the uterus itself because you're strengthening the uterus. You're also producing more blood vessels underneath of it so that you're getting ready in case a pregnancy occurs. After, when the egg follicle is released, and say for instance is waiting for the sperm cell to come, you will then enter what we call the luteal phase. The previous phase is called the follicular phase, and this one is now called the luteal phase, in which case the progesterone level is maintained at a very high level in case of pregnancy, because you need to have a stronger muscle mass to hold the growing embryo, and which later becomes a fetus after 8 weeks. This process will be maintained at such a high level along with the level of estrogen um, in case of pregnancy. But as you can see from the trend that you see in this graph, if the egg cell itself does not get fertilized, well, then you no longer need to maintain the thickness of the uterine lining. So then you kind of let it go so that you would um, be shedding off the extra layers of this extra um, lining that you have created over the last couple of weeks of cycle so that you will return back to normal. You still have a strong muscle there, but not extra strong in case of pregnancy to occur. And as I said at the beginning of the lecture, I will be sending you a separate graphing activity to track and follow to see how these things changes. Now, what are some of the methods? Oh, by the way, oh, here's a diagram that I found that I thought was interesting. Now, you often do not see this on an exam. However, what I wanted you to do was I wanted you to compare and contrast how the different hormones involved in the menstrual cycle changes with and without contraception, which means here is the lining that you see, and you just saw this, right, in the previous slide, when the egg cell doesn't get fertilized. But what if it does? And now you have a zygote that is formed. And then the zygote is implanted into the female's uterus, which is what you see over here. You see how all the capillaries are growing towards it and supporting the new life. Well, and if you take a look at how these hormones changes by all these different color lines, you will notice how much the hormone plays a role in maintaining the pregnancy and making sure that the baby will survive through this period of time in the duration of 9 months. Now, by looking at this graph, you can also ask a number of very interesting questions. One of them is, what if I don't want to get pregnant because it's not the right time? Well, that then becomes a contraceptive method. So one of the ways in which a chemical method in which contraceptives work is that women may take synthetic female hormones called birth control pills. And what that really does is that they prevent the release of egg cells from the uterus. So from the ovary, I mean. So essentially, you leave the ovary intact inside the follicle. You don't let it become released, and you don't have one waiting in the fallopian tube. And so that's one way to prevent it. There are also new methods that are known as Plan B these days, where let us just say somebody accidentally had sexual intercourse with someone and they are really concerned about possible fertilization of their egg cell and they want to prevent the pregnancy. We also have methods like that too. But those are not what we call birth control. Those are not your typical birth control pills. Those types of interventions are much more restricted. Anyone who needs to buy any type of these pills actually have to show their identification and in order to get the pill itself. Now, let me show you other methods in which science have developed ways to limit fertility. Let's begin by looking at males, all right? And we'll also explore the females coming up next. There are forms of contraceptions that men can use to achieve sterilization or limit fertility. Here are the two ways, and one of the ways in which male would do it. It's by cutting or tying up the vas deferens. Now, if you look at this particular diagram, you remember how I shared with you in the earlier parts of the lecture that males have two vas deferens, one from the left, one from the right, each stemming off from the epididymis, which is where each one of the respective testes are positioned. You can either tie both of these tubes, and tying basically means that even though you are producing sperm cells on a regular basis and they're maturing normally, you actually do not allow any of these sperm cells to exit out of the penis. All that you're allowing to exit are actually the three fluids that we have talked about earlier, coming from the seminal vesicle, prostate gland, and the Cowper's gland, also known as the bulbourethral gland. So you will not be able to get anyone pregnant. However, if you are extremely concerned about getting someone pregnant or that you're absolutely sure that you do not want a child at all, you can take a more extreme and a more invasive form of surgical procedure known as vasectomy. In vasectomy, what the surgeon does is they would actually tie and then cut your tube to ensure that there is absolutely no chance that fertilization can occur. But having said that, there has been cases that I have read in the past where the tubes would heal, reconnect itself, and sometimes these little ties that are made inside around the tube can actually loosen over time, in which case you wound up having a child with someone else, even though that was not your original intention. Now, similarly could be done for the female's counterpart, in which case you'll be tying the fallopian tubes. And again, there are two, one to the left, one to the right, each one stemming in reach in direction of towards the ovary. This is typically where the sperm cells, not the sperm cell, the ovary, sorry, I misspoke again, where the mature ovum would be sitting and waiting for the sperm to come in. So what happens is the surgeon can either tie these tubes over here, and then if you decide to change your mind later on in life, when circumstances are more favorable in your position to have a child, you can actually try to untie, you can actually have them untied. And in this case, it could be done in these, like as shown in these diagrams, or you could have them permanently cut. And then these are two of the physical methods that are used respectively for male and female. Now, there are other forms of barriers. For example, use of condoms is another effective way, so long as you also combine it with uses of spermicides, where you would prevent live sperms from accidentally making its way into the female body, but hormonal methods like the one I mentioned earlier, where you control the release, the prevention of the release of egg cells. In your textbook, you have a similar diagram like this where they go through some of the actual names and forms of methods that people use. What I would like you to do is to answer the last question on the lecture note sheet about how these three major techniques work. Now, you could use some of the information that I have just shared with you and combine it with some of the things that you read from the textbook, just to give an overview and have to have some idea in order to complete this area of knowledge for yourself. Okay, this concludes this particular section of chapter 41. I will be creating another one for a slightly different topic related to this unit as well. All right, I look forward to talking to some of you soon during some of our live sessions. Thank you very much for bearing with me. Hopefully, you have found these particular video and lectures to be helpful. Take care. See you soon.